Hello, everyone, and welcome to today's webinar titled Keynote Panel Presentation Successes and Challenges Developing a 30-Second Photonic Detection Diagnostic for the Price of a Latte. This webinar is part of the Clinical Diagnostics and Research event, and I'm Susie Valdez of LabRoots, and I will be joining you as your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using the new live chat feature, joining in our presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, just simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located at the abstract window below the presentation window and follow the process to obtain those credits. Without further ado, I now want to introduce our two speakers. First, joining us on audio today is Asma Mirza, the Chief Executive Officer and co-founder of Steradian Technologies, MSc-GH Global Health at Duke University and Duke Global Health Institute. And joining her on video is Tran, the Director of Development and Clinical Affairs at Steradian Technologies. For a complete biography of our speakers, please visit the Biography tab at the left of your screen. Welcome, ladies. You may now begin your presentation.
Thank you so much, and thank you for having us. We're really excited to talk about, essentially the process of testing, where we go from bench research to a fully developed device. So we have been developing a 30-second photonic detection diagnostic, and our goal was to get it down to the price of a latte. So today, just for an overview, we're going to go through the problem and our solution. We'll dig into the science. And then something that's really interesting is when we do look at something that takes into account a lot of the hard sciences, in order to translate it into diagnostic technology, you have to have equity and human-centric design in mind. So we'll discuss a little bit of that. And as you can see from here, we hope that you're able to understand the trial and error process in developing a new device, the breakdown of designing and testing an equitable technology, and then navigating the advantages and disadvantages of POC diagnostics. All right, so what is the problem and how did we begin this? Essentially, we looked at the diagnostic market and we saw that it was really large, it was really resistant to change, and there were certain things that just were kind of keeping a backlog, and it was really noticed during COVID. So things would take a lot of time. You had one test for each disease. So there wasn't one platform where you can kind of come on and be able to see different diseases. And then there were a lot of steps. You had labs, you had people, you had somebody running back and forth taking blood samples. So it wasn't as easy as we would envision medicine to be in 2021. So what we did was we developed a device that can do breath fingerprinting, and it uses photonics. So it takes about 30 seconds from the time you insert a disposable cartridge, and you can see that on the right-hand side. We have a cartridge for SARS-CoV-2. We're developing one for H1N1 and one for lung cancer. And the device on the left-hand side is the photonic device. And really the differentiating portion is that it's multi-use. There's no assay in here, so there's no wash step. It takes one step to do all of it. You get your results immediately. Not only do you get them immediately, but you can kind of tell numerically what the viral load concentration may be. And then it's low cost, easy to use, and it's completely portable. But we were not always at this point, and I'll tell you kind of how we got there. So what we were doing as a company was we were creating human super sites. And how did we end up in photonics and diagnostics is the question here. What our device would do is it would take any external thing that a person was looking at if they wore these glasses, and it would project it back onto their retina, onto the back of their retina to be very specific. And we were planning on using it in surgery, in aerospace exploration, and then the pandemic hit. And we saw that there were so many different points of pain when it came to rapid testing. And so what we did was what we previously looked at. On the left-hand side, you'll see it says OLED display. That was the display that would take any external source and then project it onto the back of the human retina. So it was photonics that was using very discrete focal points to be able to kind of hack into human augmentation. And we took that and we used the same exact device to create rapid diagnostics. But now the issue was, where we had an OLED display before, we now had to introduce a biological component that would be able to react, fluoresce, and then be able to hit a detection system or a photomultiplier tube that would be able to amplify it. So really, our bread and butter was the engineering, but now we were like, how do we get back to the bench, to the lab, and actually start implementing, verifying, and validating the biological components that we'll need to test? So that brings us to the science. And this was quite a long road. So if you think of a basic testing life cycle, at the very, very basis of it, you start off with validation, you go to verification, you might go back to validation again, come back to verification, swing in that area for a little bit, then you go to feasibility and design. From there, you're just reiterating over and over and over again, and then eventually you go to development. And this cycle is nonstop. There's no end point, really. And in the pandemic, this cycle, I will tell you, was not this streamlined or this linear. We would go from validation to feasibility and design to verification to thinking we're at development, realizing we have to go back to reiteration. And so what we ended up with was a very, very simple schematic for the purposes of this presentation. We decided we would insert, as you can see on the left-hand side, a disposable biosensor cartridge. The goal was to have a molecular beacon or an aptamer that would fluoresce once it reacted with a specific spike on COVID-19. Once it reacted, we would irradiate it and it would fluoresce, and all of the photons would bounce off into our proprietary mirror, which would cause them to come back and collect at the controller. And this would all happen at the speed of light, which was great. And then you'd have a quantitative output. So from the engineering side, we were golden. We were pretty confident on how to move forward. But on the biological side, what we realized was we wanted a requirement where we didn't have a wash step. We didn't want to add any additional steps. We needed to be able to distinguish from bound tags to unbound tags. So if you're thinking of a molecular beacon, which I'll show you in a minute, it generally has a quencher, and once it binds to a very specific target, only then does it fluoresce. So we needed to be able to have that differentiation in wavelengths. And then we wanted to use fluorescent activation, wavelength shifts, and photon emissions to really hit this home. And on the right-hand side, you'll see what we were trying to avoid, which is the multi-steps of an ELISA test, the multi-steps of also PCR tests, which really are the gold standard, but when it comes to public health, they do take a long time. So what we wanted to do for the RUMI was collect the sample, irradiate, and read the results. And so the plan was to have something pre-reaction, an aptamer bound to an antisense or a molecular beacon that had a fluorophore and a quencher, and upon seeing the target protein, they would break apart, the aptamer would have higher affinity to the protein, and then the fluorophore would be excited. And luckily enough, we were able to accomplish what you see on the left-hand side, where we compared our probe to COVID-19 and SARS, and as you can see, the binding to COVID-19 was beautiful. So that's what we had on the biological side. And on the hardware side, what we wanted to do was make sure that truly, indeed, we could focus what we found in breath condensate and be able to have that discrete focal point and get a good limit of detection. So we were able to get about 500 infectious units in a sample. And for COVID-19, you generally have about 10 to the 9th in alveolar air because we're using breath samples. But this was not an easy road. Here you can see we were testing it against several different variants. There was a high level of cross-reactivity. We're still kind of working through a little bit of this. When you think of cross-reactivity in other areas of the world, it might be a problem because MERS is still around, but in the United States, we don't really see it as much. So we're pretty confident that our aptamer, when it does bind, it would be to COVID-19. And that's kind of where we were in the beginning. We were just testing. We would see a lot of aptamers. We had started off actually with a molecular beacon, and the molecular beacon was not binding properly. There were a lot of kinks in the hairpin loop, and then we decided to switch over to doing a sense and antisense probe, and that ended up working much better for us. And here are just some of the results that we had from the first time around. As you can see, what aptamer complex 1 is the aptamer that's bound, and aptamer complex 2 is another aptamer that's bound. Then we put it in with COVID, and what we saw was a very, very slight reaction shift. So we found an increase in fluorescence in the presence of the protein and increased quenching in the presence of the protein, which is not good. And we thought maybe it had to do with some contaminants. The first time around, we did it in water, then we switched over to buffer. So it was a lot of trial and error while at the same time we're still developing the hardware. And again, you can kind of see here, you know, the quenching was pretty high, and that's not what you want when you're trying to get an aptamer to bind pretty specifically to a target of choice. So now moving on to the hardware, one of the trickiest parts was condensing the aerosol particles. So when you have somebody breathe in, what we needed to be able to do was take their breath and condense it into a condensate that would then react with the aptamers in at the bottom of the tube. So we had to use a TEC controller and a thermoelectric cooler to be able to really take the breath, cool it down, and create a vapor out of it. That was one of the coolest things that we've done on the breath side of things. We had to do a lot of nebulization work in the lab, but now we're able to collect breath using our device, and we're actually about to partner with another initiative that is going to be using our breath collection for testing another respiratory disease. So there's a lot of different aspects just using the device itself, but for us, we really wanted to move forward with COVID-19, be prepared for this pandemic, and be prepared for the next pandemic. So that's on the air side. Then we had a lot of issues with filtering the light. We had to set up collimating lenses, we had to have filters, we had to have them at several different angles and make sure that the way that the light was bouncing off to get to the photomultiplier tube was in such a way that we were not losing any of the photons that we collected. And that took, I would say, about 2 to 3 months of just trial and error back and forth with our electrical engineers, with the mechanics portion of it. So a lot of it was just printing, 3D printing and reprinting and just seeing what fit and what didn't. It was a lot of hours in the lab. And then something that we actually got from our eyesight recreation device was edge detection. So in the eyesight recreation device, we wanted to be able to have somebody really be able to see where the edges were. So we took that from our software, and now we're able to use edge detection to make sure that we reach that threshold of the sample that will be standardized across every single person that blows into the tube. So it's about 15 microliters, and you can see up on the right-hand corner an actual photo of what that looks like. So once you blow into it, the thermoelectric cooler starts. The RUMI device will actually start flashing when you've hit that fill detection. So a lot of really cool and innovative things that we were able to do along the way because we had to go back to that reiteration process and then go back from reiteration to validation to verification. So it's been pretty fun. And now we're at the point where really we're looking at the reiteration, and I'm really excited that Charles is going to talk to you a bit more about this, because for us, the reiteration has to do with the human-centric design. So the biological components, the workflow, and the actual optics of the device work, but will they work when it's out in the field? And what you see in front of you is essentially the model that we think we will use for production. It'll have micro displays, it'll have a custom controller board that will replace a lot of the things that we have in the lab. We need custom battery packs because it's very important for us that this will be portable. And then we want to have a trigger to start the test, and that'll irradiate the substance, it'll start the thermoelectric cooler, etc. And so the RUMI workflow is really supposed to be 4 steps, where you select the cartridge of choice, you insert it into the device, the patient blows into it, it'll indicate when they have enough breath sample. Once it has enough breath sample, it'll start the test, and it'll display a positive or negative in 30 seconds, but it'll also give you a quantitative concentration of the pathogen load. And then it can seamlessly send this information to any network of choice. So we have a software system that can help you connect, whether you're out in the field or if you're at work or at school. It will be able to help you connect to those networks with completely de-identified data. So right now we are finishing up with breath and water. We've validated the use of the RUMI in those 2 samples, but we really want to move forward to saliva, sputum, urine, and blood. And as you can imagine, they come with their own host of trials and tribulations when you're in the lab. Specifically, we may need to add 1 step, and that doesn't really go according to what we've wanted, which is to keep this single step. But sometimes when you're innovating, you do need to take into account that the first iteration may not be exactly what you want at the end or what you'll be using in the field. So especially for something like saliva and blood, we'll have to isolate different portions of the sample to be able to see how we can really make the most use of it. So overall, we really believe in photonics for healthcare solutions because it helps with the amplification, which causes you to work at the speed of light. You can make things single step. There's a lot of versatility in applications. So because you're doing the amplification and detection via photonics, you can use this in a host of things, whether it's checking water for environmental safety, to checking chemical and biological warfare in air, to looking at diseases in breath, whether they're communicable diseases or non-communicable diseases. It allows you to have portability. You don't need a lab to prep anything, and it's very inexpensive because you don't have reagents, you don't have buffers, and at least you don't use as much as you would in a general assay. But again, the disadvantages are that you have 100,000 iterations. Novel technology requires adoption. So before you go in and you have this amazing idea, you do need to be able to look at, once I'm out of the lab, what does the commercialization of this look like? How will people be convinced of the fact that we're doing this amazing science, but it'll translate to real life benefits for them as well? And then there are a lot of regulatory hurdles. If you think the testing life cycle I showed you earlier was complicated, the regulatory life cycle is 100 times more complicated because with anything that's new, you have to have the burden of proof to be able to show that we are not using lasers that will blow through somebody's mouth. And so a lot of common sense things, you really have to kind of lay it down, write it out, and be able to defend the technology and be able to have your data ready.
Thank you so much, Asma. And I think that a lot of the science that you guys have been able to do and that we've been able to do so far is really interesting. I'm going to talk about what it means to design for human use and also with equity in mind. So first, I do want to acknowledge, like Asma did, that this process is not linear and that a lot of times you're going to have to go back to stages that you thought you've already accomplished because you're going to run into a problem that you just really didn't anticipate. But in general, these are kind of these 6 high-level steps that you need to take an idea or innovation and get it at least in front of a person. So first, you have to have an idea. For us, that was developing the RUMI after we pivoted from the eyesight technology. Then you have to experiment and establish a proof of concept in the lab. That was a lot of the science that Asma just went over, and it's really, really important for you to be able to develop a working prototype that you feel comfortable using with people, because then your next step is establishing a proof of concept in the clinic. This is where you're going to start doing research with human subjects, and is also where you now are entering an entirely different realm of regulatory procedures and rules. After you have a working prototype that you feel very comfortable giving to somebody else, so you don't need your team of engineers or researchers or designers at hand, then you can start really engaging with the FDA to develop a regulatory pathway, and you can begin conducting your clinical trials. I think that a lot of these steps are really important, but stage 4 is crucial for you to think through before you start, because you don't want to waste your time or your money conducting a clinical trial that maybe you don't need, or developing data that may or may not be sufficient enough for the regulatory body you're submitting to. After you finish all of that awesome work, you then have to develop and implement a quality management system for your technology. At this point, you need to have design freeze, and you need to have already figured out who your subcontractors are, manufacturers are, etc. At this stage, you will also begin achieving your FDA approval. Once you do get that and you have your quality management systems in place and you have your manufacturing in place, you can then start doing the other nitty-gritty work of getting your technology into people's hands. Granted, every single market requires a different regulatory approval process and market strategies. So you definitely start with one and then move forward. However, it's always crucial to innovate with purpose, and it should be driven by real-world problems that need solutions. For us, when we were developing the RUMI, we saw COVID-19 and rapid point-of-care diagnostics as a huge and critical need for not just us as individuals, but for the global world and society. We really believed that our technology could really fill an important need, but we weren't 100% sure, which is why we really wanted to talk to end users, so people who would be administering the RUMI or people who would be using the RUMI on a daily basis to better understand whether or not our technology can actually accomplish our goal, which is solve a problem. So a great way for you to incorporate end users early in the design process is through conducting these interviews. And this is kind of a process that should be iterative and done throughout your entire design process. This is a way for you to really begin doing human-centric design, because you're incorporating the people who are going to use it at the end of the day very, very early on. We really believe that understanding how our technology will be used is how we can better design in the future, not just for our COVID-19 prototype and COVID-19 application, but for lung cancer and other diseases in the future. However, we have a lot of challenges, like everybody else does, when it comes to bringing novel technology to the people who need it most. For us, we've run into challenges around finding funding, around developing and getting data, around a regulatory pathway, and executing clinical trials. Our first hurdle is a catch-22 that I think a lot of you might be familiar with, which is you really need data to get funding, but you really need funding to get data. There's a lot of ways that you can work through this problem, such as getting grants, collaborating with other institutions that might have more money, or finding investors. And while all of these options do have benefits, they all have different kinds of consequences. So it really depends on not just your innovation, but your level of support. If you're able to get past this catch-22, you're then going to be faced with these critical decisions like developing a regulatory pathway. For us, we're really trying to decide and understand whether or not a de novo process with the FDA is better than a 510K, or whether or not it's better for us to incrementally apply with the FDA or do it all in one go. Each of these different pathways will change not just our development, but also our market strategy. So I highly suggest that you try and talk to as many experts as you can in regulatory science before you really start pushing this stuff through. Another thing that you really have to think about before you engage in clinical trials is your ultimate use case of your innovation. So when we're talking about point-of-care diagnostics, we're talking about who is going to administer it, who is going to do the data collection, who will need to process it. Will this be used for diagnostics or will this be used for screening? Are you going to use it at home? Are you going to use it with children? All of these different use cases require specific safety and efficacy data for, again, different regulatory bodies to review. So while some technologies might not require a lot, such as like a new tongue suppressor or something like that, I would say the more invasive your technology or the larger the claim you have, the more data you will need to be able to prove to people like the FDA that your technology is safe and providing an accurate diagnosis or screening. Another thing we really want to think about is designing with equity in mind. And for me, I really like to use the World Health Organization's definition. For them, equity is the absence of unfair, avoidable, or remedial differences among groups of people, whether those groups are defined socially, economically, demographically, or geographically, or by other dimensions of inequality. Health is a fundamental human right, and health equity is achieved when everyone can attain their full potential for health and well-being. I think that this definition is great because it's really expansive in terms of what we think about when we're thinking about differences, but it also is very cognizant of the fact that we are different in general as people. When we think about health equity in designing new medical devices, for us, we really tried to think about diagnostics in terms of stuff that's too expensive, getting a diagnostic that might take too long or it's too far away for you to get to. It might take too many steps, so there are too many follow-ups for you to deal with, and also trying to combat stigma of diagnosis. How we did that was through a variety of things, and they really kind of point to these three critical aspects of health equity. We want to try and increase access, both physically by making sure our device is battery operated and portable, but we also want to make it accessible economically. So we're trying to make sure our tests are less than $3. We want to increase access by eliminating the need for follow-ups, so we want to make it one step. And because we are able to have the RUMI connected to telehealth, it will hopefully allow for easier and faster diagnosis in places that may or may not have health clinics. Again, we also want to design with stigma reduction in mind, because we recognize that being screened positive for something, even like COVID-19, comes with social things. A part of the beauty of the RUMI is that it is small and discreet, and that a lot of the screenings or diagnostics can be folded into regular health checkups. We also believe that the oral administration of the RUMI will help decrease stigma by not leaving any signs of testing left over. We also believe that we want to increase equity in point of care diagnostics, because accuracy is critical for the continuum of care and the development of future health plans. And we believe we can do that by eliminating the lab and reducing human error, and providing a qualitative and quantitative results almost immediately, allowing providers to understand not only if their patient is positive or negative, but also hopefully where they are in their disease spectrum. Granted, there are always unintended consequences when you're developing new technologies. And when it comes to point of care diagnostics, it's really critical for all of us to think about what is our role in the continuum of care? Like what happens after your device tells someone they are screened positive? Do you need to provide information on treatment? Do you need to ensure that these results get to a particular body, such as the public health department? A lot of these questions are not necessarily things that we will think about very early on in the design process, because it takes so much just to get your technology to people. However, things like unintended HIPAA privacy violations will come into play when you're trying to get FDA approval. So it's always good to think about it early on. Another really important aspect of innovation is ensuring that the technology we develop does not increase inequities that currently exist. And one of the things that I think we've really seen recently is this reliance on telehealth, especially because of COVID-19. However, telehealth requires people to not only have an individual device, but for that device to be able to conduct a HIPAA-compliant appointment, which requires strong and consistent internet. You also need a room or a space where you can talk to your provider alone, which is not really possible for a large proportion of this country, let alone the world. So when we're developing technologies, it's always important to remember that not everything can solve social problems. So we need to be cognizant of those social problems as we move forward. So thank you guys so much for listening to us. I just want to go over a little bit of where we at Steradian Technologies are now. We are the grand prize winners of XPRIZE's 6 million rapid COVID-19 testing open innovation track competition. This has allowed us to begin our initial feasibility study, where we are doing device integration, which means we are integrating the RUMI handheld device and the disposable biological cartridge. We're also conducting a feasibility study, which is our first study with human subjects using the RUMI. And we are developing a regulatory pathway with different consultants and regulatory scientists. We are also, because we are able to work as a platform detection device, working on detecting early-stage lung cancer with Johnson & Johnson, and developing a multiplex with COVID-19 and H1N1, and hopefully in the future, the entire flu spectrum, with La Jolla Institute for Immunology and XPRIZE. And we also are having very interesting and exciting conversations about using the RUMI's breath concentration technology in TB. So we're super, super excited about all this stuff that's to come. We hope that you guys enjoyed our presentation and were able to learn about these different things in terms of what it takes to go from ideation to innovation, how you can design with people and equity in mind, and what are the advantages of point-of-care diagnostics. Lastly, we want to say thank you to everyone on our team, everyone at LabRoots, everyone at XPRIZE, and of course, our incredible scientists and innovators. Thanks.
And thank you both for that informative presentation. We'd like to now start the live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in. Our first question, how would multiplexing work?
Sure, I can take that question. This is Asma. So the way that multiplexing would work is on the device side, we would need to have different filters that could filter out different wavelengths, and then be able to bring them to the photomultiplier tube and be able to differentiate which wavelengths are coming in, because we would still want to know the relative concentration of each one of those biomarkers or pathogens that we're looking at. And then on the biological side, what we would need to do is have different molecular beacons with different fluorophores attached to them to be able to differentiate. And then it would be very important to make sure that there isn't any cross-reactivity in the biosensor itself. So it would definitely be a multi-step process, but it can be done, which is very exciting.
Thank you so much. And how would you expand into other samples?
So when we think of other samples on the device side, not much would change. What would likely change is on the cartridge side. So if we're looking at blood, when you take a blood sample, you may have to isolate it into its different portions and then pipe that out the layer that you're looking at, insert that into the RUMI device, and then look at it. So that's just a very high-level way of thinking about it. But, you know, as innovators and as scientists, we definitely believe that there's something there that we haven't hacked yet. But I think expanding into other samples, just strictly from a testing life cycle stage, we would have to do a lot of in-lab testing without even using blood in the beginning. We would just have to figure out different ways that we can isolate things. So it would again a multi-step process. All of these would be multi-step processes.
Thank you so much, Asma. And I want to thank our audience for these great questions. Where do you see your device being adopted in screening or diagnostics?
Hi, I can definitely take that one.
Yeah.
Okay, so it really depends on what disease we will be looking at. I think for things like lung cancer, we're really looking at screening. In terms of COVID-19 at home use, it would also be screening, but COVID-19 in the clinic would be a diagnosis. So use case also really matters here in terms of where it would be adopted. But we do believe that because it's a platform device, it would be able to fit well in any primary healthcare physician office.
Wonderful, thank you so much. And we have a couple more questions coming in. Was your pivot intentional, and how early in the development was your original device?
That's a great question. So the pivot was not intentional. We were very, very excited to kind of be in the space of deep tech, and the recreation of human eyesight to just human supersight would help for anything from retinal degeneration to better accuracy in surgery. And so we had started our preclinical testing and studies for that, but when the pandemic hit, we had to pull back all of that because nobody was allowed to go into the hospitals around, March 2020. Everything had to cease and halt. And we were just very serendipitously talking in a meeting, and we were discussing the lack of public health initiative when it came to rapid diagnostics, and we kept thinking, why aren't they fast enough? And as we dug through that question, it dawned upon us that, hold up, what if we use our device to maybe use photonics for diagnostic testing? And that's how it came up. So we didn't even have a device. We just had the internal mechanisms of eyesight recreation, and we started from, a blank slate. So I would say we weren't even early in development. This was nascent.
Thank you so much. We have one more question coming in. Is there a difference between communicable and non-communicable diseases?
Yeah, I can answer that one. There is a difference for us in terms of how and what we are going to identify using the RUMI for communicable and non-communicable diseases. I think the biggest differentiator is that with communicable diseases, we're more than likely able to look for the disease itself, whereas with non-communicable diseases, we're looking for the way that the disease will affect the body, if that makes sense.
Absolutely. Ladies, I want to thank you for your presentation today. Would you like to provide any closing remarks before we go?
Yes, we are so grateful to be here and to engage with a community that innovates and tries to make the world a better place through science. And we're always looking for scientists who are interested in technology such as ours. We love collaborating, so please feel free to reach out to us.
And Tra, do you want to add anything?
No, I think Asma totally got it all. Thank you, everyone.
Perfect. Well, thank you again, Asma and Tra, for your time today and for your important research. And before we go, I want to thank our audience for joining us today and for their interesting questions and questions we did not have time for today, and those submitted on the on-demand period will be addressed via the speaker using your contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years up till November 2023. Labyrinth will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and stay safe. Bye-bye.